Okay, so we're moving into chapter 5, and 5 is the end of this initial unit where we build upon the basic organic chemistry that we've talked about. We build upon our functional groups that we talked about in chapter 4. Chapter 3 obviously got us into issues with polarity. Chapter 2 was just basic chemistry. So it all kind of comes together with this. And a lot of this material is a refresher of what you would have had in an honors bio course, but we'll obviously go a little bit deeper on some of our details just to make sure that everybody is in good shape. So when we start looking at our categories of organic molecules, the 4 large classes, we're talking about the carbohydrates. So that's why I had that lab yesterday, just kind of getting us thinking about the different categories within the carbohydrates. We have the lipids, which are a unique group because they're kind of a collaboration of a bunch of different types of molecules that are maybe not as similar within that group as the carbohydrates, but they definitely have some similar properties. We have the proteins, very critical. We want to absolutely understand how proteins are built and how the genetic information flows from the nucleic acids to the proteins. So one of the common themes that goes throughout this whole chapter is the idea that we can start with monomers. Okay, monomers are going to be small molecules, and we can bond them together, and we can build macromolecules, which in certain instances they can become quite large. They can be very extensive. So the idea is that there's a definite connection between the structure and the function of what we're going to be dealing with. And I think we've already kind of addressed that even as we've gone through and we've talked about different isomeric forms where maybe one drug has the enantiomer mirror image of another one, and one is effective and one is completely ineffective. So that theme kind of carries through, and we're definitely going to see a connection between structure and function. So just making sure that we are clear, section 1 really just starts about building larger molecules. It's just the theme that we are going to see bigger molecules. So a polymer, poly, indicates it's going to have many parts. So it's going to have many of the similar building blocks that are going to come together, and those building blocks are monomers. Now, what we're going to find is that we had 4 groups, but 3 of them technically will follow the monomer building up to the polymer protocol. There's one group in there that doesn't follow that protocol. That's the lipids. Okay, the lipids, as I kind of indicated, they're a collection of things that maybe don't seem as connected as like the carbohydrates. But the lipids, they definitely increase in complexity, but they don't have a true monomer building polymer strategy, I guess you could say. All right, so those ideas, if we take a look at it, one of the things that we need to understand is that there's definitely going to be 2 overriding processes that are going to be associated with synthesis and breakdown. So if you're looking at synthesis, you're building up simple into something more complex, and then obviously if you do breakdown, you're going in the opposite direction. And there's energy transformations that take place with these as well. So our first one that we have is a dehydration reaction. Okay, dehydration reactions are whenever we put 2 simple things together, and when we do that, we get a more complex molecule at the expense of losing water. So it's easy to think about, when you have somebody who's dehydrated, they've lost water. A dehydration reaction is going to involve the removal of the water. So way back in chapter 3, I asked you a question about sucrose, and I don't think I was able to trick you on that because somebody had indicated that while you're taking glucose and fructose, when you chemically bond those together to make a more complex molecule, you had to remove water. So anytime you go simple and simple We put it together, it's dehydration. It's also sometimes referred to as a condensation reaction, okay? They're both the same thing. So condensation reactions, dehydration, synthesis, they mean the same thing. Now, if you have something that's large, you want to go in the opposite direction and break it down, you do the opposite process. They use hydrolysis. So if you think about the word hydro, you should be thinking water, and lysis is to break something. So if you have a larger collection of these individual units in the form of a polymer, and you want to break it down, you want to digest it, the idea is that you're going to be executing hydrolysis reactions. So these are two going in the opposite direction, basically. And what we're going to find is that one of them is going to be described as being anabolic, and one of them would be more towards being catabolic. Is anybody familiar with those two terms at all? Anybody know the difference between anabolic or catabolic reactions? So we will talk about this in chapter 8. It's an energy chapter. So I'm not going to get too much into the energy, but if you're building up, that's an anabolic process. So which of these would be the anabolic process? Right? We're going to be building up. All right? So catabolic is breaking something down. Okay. So we will see that. And here's the thing. In our bodies, because these are energy demanding reactions, what do you think we depend upon, aside from just the energy itself? What makes these reactions likely to occur inside of a living organism without having to really crank up the energy level? What do living things rely on? What do we as humans heavily rely on to make all of our biochemical reactions possible? Yes. Very good. So we rely on enzymes, which are biological catalysts, right? So a catalyst is something that is going to make that reaction more likely to happen. If I remember how a catalyst operates, are catalysts used up in the reaction? So a catalyst is something that is not consumed in the reaction, okay? We have a whole section where we get into the details of how a catalyst works, but for the most part, we can think that it just makes the conditions of that reaction more favorable to occur. Okay, we can kind of leave it at that point for now, and we'll talk about it later. All right, so let's look at these two images here. So in the first one, we got the dehydration, and we're going to make a polymer by synthesizing a short polymer with another component of a, here we have a short polymer, here's an unlinked monomer. So the idea is, in this scenario right here, and these are carbon chains, and we have a hydrogen sticking out at this end right here. Over here, we have an OH. Okay, we talked about that as being one of the alcohol groups. When we bring these two together, we are going to release water. Okay, so that's a condensation reaction or dehydration. So that builds up something that's going to be more complex. And as a result, it's going to be energy consuming to do that. Now, if we go the opposite direction, we have something that is already a polymer, and we wanted to break it down into smaller units. So maybe I want a section 1 through 3, and then just have this one individual. So by adding H2O, we add a hydrogen here, we add an OH here, and that's a hydrolysis. So if you're breaking something down, obviously you're going to rely on water. There is going to be, the condition of this new state is probably going to be of lower energy, so there will be a release of energy coming off of it. And that's typically what we would find with catabolic there. All right, so basically what we're going to find out is that when we look at our cells and we start to think about what is inside of our cells, we've got building blocks, and then we've got molecules that are more complex, and they've built up over time. So every cell is going to have lots and lots of these different macromolecules that are going to be, populated within that overall cell. Okay, and what we'll find is that different cells, even within the same organism, are going to have different populations of macromolecules. So as a result of all this, we get a tremendous diversity of molecules that we can work with. All right, so that's just kind of the really, really general introduction. So what we wanted to do for today is I really want to jump into the heart of the organic molecules by jumping to the carbohydrates. That's our first one there. So before we go too far, we know carbohydrates are sugars, right? That's kind of the scientific way of referring to our sugars. We call them the carbs. So what are some properties that we would be familiar with in regards to discussing carbohydrates or sugars in general? How would we classify? What would be the function in a living thing? What's the function? Give me one function of a carbohydrate in a living thing. Yeah. Okay, so right off the bat, one of the first things that we recognize is that these are energy molecules. They're going to have hydrocarbon chains, okay? So they're going to be an energy molecule. Do we consider sugars to be a long sustaining energy or something that is relatively short in duration when we look at other food sources? So we'd probably consider the sugars to be something that's kind of like an immediate energy source. In fact, you take a look at some of the energy drinks, what are they? They're loaded up with carbohydrates, okay? They're sugars. And the idea of those sugars is that they're going to provide a very short range boost of energy, which may serve a certain function, but depending on what physical activity you're doing, you might need something that's more sustaining. So we'll see that there's other categories that'll be a little bit better as far as supplying that. So these are fuels, and these are also building blocks, okay? Where else in our cell, besides from the energy perspective, can you think of anything else that a carbohydrate would be responsible for in our body? Yes? Storage, okay. So we definitely have capability of storing the carbohydrates and mechanisms of building up simple carbohydrates into more complex ones. So it serves, like in our case, we use glycogen as an animal. We store that in our liver. That would be a more complex carbohydrate. Whenever you take a look at the cell membrane, there's carbohydrates that are on the surface that in many cases act as markers and identifying characteristics. So even when you think about how cells recognize one another within a living organism, or how our immune system recognizes certain cells, part of that is markers on the surface that include carbohydrates. So the carbohydrates are the sugars, and they are going to be made up of everything from the simplest sugars, which we call monosaccharides. So the naming sequence is pretty easy. Mono means one. Anytime you see saccharide, you should be thinking sugar. So the single sugars. So, the main ones that we would see for that would be like the glucoses and the fructose and the galactoses. Those are all monosaccharides. And the idea is that those monosaccharides can build up into a diversity of different types of polysaccharides. So out of the stuff we looked at in our lab, I know that we had sucrose. I know that we had glucose. I think we had fructose. We had lactose, and we had starch. So we actually have 3 levels of complexity within the carbohydrates represented in that chapter, or in that lab, okay? All right, so here's the general formula for a monosaccharide. They all are pretty much built the same way. So they're going to have a 2-to-1 ratio between the carbon and the hydrogen and then the oxygen in most cases. And, a lot of times we see C6H12O6 and we immediately say that's glucose because glucose is the product of photosynthesis that our plants are going to execute in so many ways. But we also know that there are different isomers of C6H12O6. So if you take a look here, we will see that fructose and galactose are also C6H12O6, but they have a different configuration. So one of the things that we are looking for is where are the carbonyl groups. So you might recall from yesterday and from our discussion, we have a carbonyl as a C double bonded to an O, and if it's going to be a ketose, is that double bonded C-O interaction, is that inside the molecule or is it at the edge of the molecule if it's a ketose? Ketose is going to be inside. Key goes inside the lock, right? Ketose is inside. Where's the aldehyde? On the outside, right? So the aldose will have the carbonyl group on the perimeter of those two things. So if we look at a couple of different categories of sugars, we'll see how that slight positional change impacts what the sugar's properties are going to be. So let's look at this first one here. So these are called trioses, and the idea of these are that they're going to have a 3-carbon sugar. And if we go through here, this one's referred to as an aldose, which is an aldehyde sugar, and the carbonyl is on the outside. If we look over here, this is a ketose, and it's in the center of the molecule, all right? So it all just has to do with the position of that. I'm not going to ask you to memorize like which sugar is which in this case right here. I'm just trying to get you to recognize that the position of that carbon double bonded to an oxygen is going to be an influential factor as far as what category of sugar we're looking at. So these are pentoses. These are 5-carbon sugars. Okay, ribose. We'll definitely be talking about ribose sugar later on throughout the course. We can see that this is at the edge, so that makes it an aldehyde, also an aldose. This one right here, ribulose, it's interior. It's a ketone or a ketose sugar. Okay, so it's just recognition of those things. So the sugars that we tend to spend the most time talking about are the hexoses. These are the C6H12O6s, all right? And if you look at the chain form of glucose and galactose, they look nearly identical. They are aldose sugars, but the only difference is there's a flipped orientation between the alcohol and the hydrogen group on the glucose and the galactose. The one that stands out looking significantly different when we look at the ring structure is fructose, okay? And usually, for the most part, we tend to look at things whenever they are in a ring because that's typically what's going to end up happening whenever they're in a water-based environment, which is what we see for most of those things, okay? So fructose, when we look at the ring structure of fructose, we're going to see that it's going to form a 5-sided. So always think fructose 5. Even though it's C6H12O6, it forms a 5-sided, like a pentagon structure, whereas we have a 6-sided glucose and galactose. All right, so we see them linear in that picture, but whenever it's in water, they form rings. So most of the time, whenever we are talking about our, our structures of the different carbohydrates, we usually look at them from a ring standpoint. So I'll show you how they kind of form those rings and some of the basic characteristics of each of those different things, okay? So we had already talked about this. We said the monosaccharides, these are simple sugars. These are simple sugars, so they're going to be a good fuel source, and they're also going to be a building block for some of the other components that we will go through in establishing an organism and building up the cellular components, whether we're talking about the membrane components and some of the other energy sources that are stored throughout the cell. All right, so if we take a look at how that linear form can be converted into a ring form, we'll see that progression on this next one. All right, so here we have a linear form, and when we number our carbons, obviously we're starting at the top and we're working down towards the bottom. So this is a 6-carbon hexose. So whenever it forms into what we would consider our ring structure, we start our numbering at this position, because we're gonna be able to identify some changes that are taking place between some of the different components. Also, if you look at this compared to this diagram right here, this is an abbreviated version because every point in here is not referenced as a carbon, but we know it's a carbon unless it's identified as something different, because that's a carbon structure. So this one, we actually go through and we label it carbons and oxygens and things of that sort. So position number 1, we have hydrogen in the up position, we have hydroxyl facing down, HOH, OHH, HOH, and then up here we have a branch that comes off of it with the 6th, and then there's a hydrogen that breaks off of that. So that is going to give us what our typical glucose configuration is going to look like. Okay? Now, we'll see some other images of this, and we'll look at some of the diversity that goes on within those molecules. Now, if you build from one monosaccharide and you build up to 2 combined monosaccharides, you end up with a disaccharide. So some of our disaccharides that we've talked about, we looked at sucrose. So if you combine a glucose and a fructose, take out a water, you have your sucrose. And you might remember when we were doing the calculation of, we were doing like a molar calculation problem with that. So for sucrose, we were taking C6H12O6 plus C6H12O6. So maybe we could say this is glucose, this one's fructose, but then we have to subtract out the water. Okay, so when we put these together, we get a C12H24, but subtract out the 2 hydrogens, so that gives me 22. And this would be 12, but subtract out the 1 there. So that would be a typical chemical formula for a disaccharide, all right? Because you have to remove the water. Take that out of there. Okay, so in this case right here, the type of bond is a covalent bond, and it's specifically known as a glycosidic linkage. So a glycosidic linkage is a covalent bond that's formed between the different monomers of a more complex carbohydrate. So what we're going to find is different types of glycosidic linkages will influence how we're able to go through there and literally be able to digest or break things down. Okay, so let's look at an example of something on this next slide here. Let's look at this one right here. So this is the combination that we were looking at in terms of forming one of our disaccharides known as maltose. And when we do this combination, here's my first glucose, here's my second glucose, and these are what we call alpha glucoses. So whenever you look at this, this is trying to depict something that's 3-dimensional in like a 2-dimensional image. So when something is literally on the upside, if you kind of think about my ring structure kind of being the plane of this paper right here. And then if I were saying, okay, my caps, we'll say the caps are the hydrogens. So we would have oxygen up in the corner, and then we would have, at this point right here, we'd have hydrogen sticking up. Over here, there'd be hydrogen sticking up. This one, the hydrogen sticking down. Okay? Over here, hydrogen sticking up. So that's going to make a difference whenever we try to combine things in a second. Over here, This one has the mirror image structure. So when you put these two together, you have an OH and you have another OH right next to each other. So when those two are able to combine, what we do is we release that water molecule. Okay? So that combination, let's take a look at this. This is position carbon number 1. This is 2, 3, 4, 5, and this is number 6 up there. Over here, position 1, position 2, position 3, this is position 4. So this linkage, it's called a 1,4 glycosidic linkage because you're combining position number 1. Oh, didn't mean to hit the eraser button, but you get it. So I'm combining position number 1 with position number 4. That's a 1,4 glycosidic linkage. Now, right now, that doesn't seem like that big of a deal, but in a few minutes, I'm going to show you that there are some differences between what we're capable of breaking down and metabolizing, and in some cases, things that we cannot break down or metabolize. Okay? Let's look at the bottom picture. This is synthesis of sucrose. We already talked about that. So here's glucose. Here's our fructose structure. Remember we say fructose, think about it, it has 5 sides to the structure. That's because of that position of where that oxygen's located at. Okay? They both are C6H12O6. Fructose, think of the 5-sided structure. Now, when I put together this setup right here and I count out my carbons, this one happens to be in the 2nd position. Okay? This is still in the 1st position. So this is considered a 1,2 glycosidic linkage as far as how we number them. Okay? So that's just part of the nomenclature and things of that nature. For this right now, all we just want to recognize is that we're combining simple and we're taking out water and we're able to end up with a molecule that has greater complexity. Anybody have any questions on that at all? Nothing should be too new with this. All right, so monosaccharides, glucose, fructose, galactose. Disaccharides, things like sucrose, lactose, right? We talked about milk sugar would be a lactose product. Some of you may be lactose intolerant or know somebody that's lactose intolerant. What do you think that means if somebody is lactose intolerant? If somebody is lactose intolerant, yes. Good. So what was mentioned is he said that you need an enzyme to break down that combination to make that into the simpler sugars. So genetically, you're not making the enzyme, which is a protein, so that you don't digest it. So if you don't digest it, either A, you don't feel real well and you are sick, or B, you have to supplement it, or C, just avoid it. So those are your options with lactose. All right, polysaccharides. So that's going to be your largest group within the sugars. And the big things that we are going to see with the polysaccharides is they tend to function in terms of long-term storage, or longer-term storage, I should say, and they play a role in structure. So the polysaccharides are going to ultimately be determined based on what their individual units are that make it up. So what are the sugar monomers that build it, and what kind of bonds hold them? So they're going to be covalent bonds, glycosidic bonds, but what's the position of them? Are they going to be oriented in such a way where they form more of a straight chain, or are they going to be oriented in a way where they give you branching? Those are all things that will show up with this. Okay. So, mentioning also in some of our monosaccharides and disaccharide categories, fructose tends to be a very, very sweet sugar. It's the fruit sugar. Lactose, that disaccharide we talked about, was in milk. Sucrose is kind of table sugar, the stuff that you bake with or people might put in their coffee. So when we move into the polysaccharides, we're going to look at them from two different perspectives. Those that are more oriented towards storage and those that are going to be more oriented towards energy or energy structures. So storage or structures, I should say. So our first one is going to be starch. Starch is going to be how plants store their sugar reserves. And you got to figure plants execute photosynthesis if you're a green plant, if you're an autotroph. They don't use all of that sugar at that instant. They store it, they build it up, they maintain that for long periods of time. So by having these storage molecules, it's obviously going to be sustaining for the plant to be able to go through there. So one of the things that we can look at later on is, if you're working with even, a real easy place to see starch kernels would be in like a potato, because it's a highly starchy substance there. But anyways, plants will store that extra starch in these granules, which are part of the chloroplast there, and those are going to be accessible as a energy source later on. So the plants can actually stockpile their glucose. So we have different variations of starch. Our simplest one is going to be the amylose, okay? And as we kind of go through there, the amylose is going to give us more of a linear configuration. And then we have a second one that's going to be a little bit different in terms of the branching. Is called amylopectin. It just is oriented based on how those glucoses link one after the next. So if we examine, here's a picture of the granules. Okay, so this overall structure is showing us the chloroplast or the plastid. These are the granules of starch that are stored inside there, so they're accumulating the buildup of the sugar. If it's amylose, that's going to be the unbranched variation of it. And some of it, because of the way in which it's oriented, it starts to form multiple branches coming off of it. So that would be the amylopectin. So the top picture is just referring to starch products. Now, can we digest starch? The answer is yes. We get energy from starch, and the way that we're able to digest it is that we have the necessary enzymes to break things down. So that kind of seems to be a common theme, whether you're talking about people that are lactose intolerant missing those enzymes, or us being able to break down things that we traditionally will consume as part of our diet. So we can break down these monomers off of this overall larger polymer and be able to, release energy stores. Now, the bottom picture is how we typically store our carbohydrates. And these are in the form of glycogens. So animals use glycogen, plants will store theirs as starch. So if you eat, let's say you eat some carbohydrates as part of a snack or a meal, and you were to test your blood glucose right away, you might see some elevation in that, but we have insulin and we have glucagon that are going to regulate our blood glucose level, okay? Part of what we will do is, if we have a surplus of that sugar molecule, that glucose, we will build it up as a reserve as glycogen, okay? And then later on, whenever your glucose levels start to drop, there's an enzyme, or there's a hormone, glucagon, that's going to break that down and release it so that we can continuously have energy molecules. All right? So amylose, pretty much a straight chain over and over again. Amylopectin, giving you the branches. Glycogen, polysaccharide that animals will use. And in our case right here, we have glycogen that is stored in reserves throughout our liver, and we also will find some stored within our muscle. Now, one of the things is that there's a short shelf life for glycogen. So it's not something that's going to last for extended periods of time. They estimate that in humans, we get about a day out of our glycogen reserves. And after that, it starts to break down. So there are some concerns with some of the more radical diets where people are completely eliminating carbohydrates entirely and creating some deficiencies in those areas. So we will rely on having access to these different sugar components there. Okay. All right. So let's take a look at how these things combine, and particularly let's look at the structural end of things. So if you look at something like cellulose, so cellulose is the material that builds wood products. You can definitely get a good perspective of cellulose if you took a piece of celery and you snapped it and you grabbed the little strings, right? That is a carbohydrate that helps build the cell walls for the plants, but it's not digestible by us. Okay, and we're going to talk a little bit about why that's the case. But one of the things that we're going to find out is that it has the same raw materials as starch and some of the other sugars that we can metabolize, but the position of those glycosidic bonds are not going to be compatible. So we're not going to be able to easily break these things down, and we're not going to be able to digest it. Okay? So if I were to look at a carbohydrate, for example, if we looked at glucose, and it mentions here that there's two different forms of glucose. There's alpha and beta. So on this side of the glucose molecule, so you figure you've got this kind of orientation. So whenever you look at something that has this position right here, the H up and the OH facing down, and then the rest of it was obviously the rest of the glucose molecule. So this would also be H up, OH down, OH, H. This is going to be H and an OH, and then you're going to have a little hydrogen here. This is the alpha version. So just know that the hydrogen up, hydroxide, or hydroxyl facing down is going to be alpha. If I switch this and I just do this orientation, so instead of having the OH facing down, and I change the orientation on that one right there, this change in orientation just at that position is going to be what makes it beta. Okay? So the beta configuration, being in the opposite orientation, is going to influence whether or not we have something that we can digest or something we can't digest. So let's look at that from another picture here. Because if you think about it, what's the dietary value of fiber? And the answer is we don't get any calories out of fiber. Fiber acts as a way of abrading the intestinal tract and improving the intestinal functionality, but we don't get the calories out of it because we can't metabolize it. All right. Let's look at what's different between alpha and beta. So this picture right here, let me clean this off. All right, so in this one right here, and that's kind of what I was itemizing on the last slide, we have the H in the up position, OH in the down position. If it's in the beta orientation, those two things are flipped, okay? And that's all that's showing us is that these are kind of reversible. So we're kind of altering and moving around in that particular position there. Okay. So if we have starch, the OH combination, this is what's called an oxygen bridge right here. So whenever you're taking an OH and the HO and they combine and they form an oxygen and then water flies out of there, that's the combination that gives you your glycosidic linkage. So if you notice in starch, everything is a 1,4 linkage. So they kind of say that the orientation is down, okay? So those are all oriented in one particular fashion. But if you try to take an alpha and a beta and you combine those things together, what you're going to find out, so in this particular situation, the beta situation, because the orientation is opposite on this side, we have kind of this flipped configuration. So the sugar is inverted every other one. So it creates a different kind of linkage. So this is called a 1,4. In that particular case, we're talking about a beta linkage. And what we find is that we have enzymes that allow us to break this down, but we don't have enzymes that break that down. So you can eat a wood product and you're not going to get any kind of nutrition out of it because you're not going to be able to digest that. Even something like a termite, we might look at a termite and say they're eating the wood, but they're not actually digesting the wood. There's a relationship with another organism in there, a mutualistic relationship where they're able to metabolize and break that down. If you were to take a look at cattle, where they're metabolizing and they're breaking down the cellulose, but if you look closer, there's microorganisms that are actually performing that function for them. So without having the enzymes necessary to break this down, it pretty much means that it's going to be non-digestible for us. Okay, so just as we kind of go through there, what we find is that whenever you have the alpha linkages, so that would be kind of what we were seeing for starch, that's going to give you polymers that have kind of a helical structure. The beta glucose gives you a straight structure. So we have the hydrogens one up, one down, one up, one down type of orientation. And like I said, the main thing to keep in mind for this is that they're just not compatible with the enzymes. Okay. And then the cellulose molecules actually get held together. So they're forming more of that straight configuration and they become very parallel, and we group these things together to build a strong building material. So if you're thinking about something that is going to be rigid, something that's going to be strong, the orientation that, whether we're talking about different fibers that would be found in, things like cotton, or if you're talking about something that's going to be oriented like the strands of wood products, the idea is that the way that those sugars are put together, it makes it much more rigid, and it makes it more unlikely that it will be digested or broken down. Okay. So if we go over here and we kind of zoom in just a little bit, and you can see how they are all clustered together, and this is building up inside of a plant cell wall. And what we're starting to get is we're getting these combinations. So they're very linear, but they're linked together, and it becomes a much more rigid structure than when we saw something like amylopectin or amylose. All right, so the enzymes that digest starch by breaking the alpha linkages are not the same. So you can eat starch, you can consume starch, but you can't consume cellulose. So that's why cellulose is fiber. It is non-digestible. It's insoluble. We do have some forms of life, as I was indicating, that do have enzymes to digest that. The organisms that would be found inside the guts of termites, things that would be relationships with, within the symbiotic interactions in the guts of the cattle, things of that nature. So they will be able to go through and execute and break those things down. But the main idea is that without those interactions with other organisms, they will not be digestible. Okay? So we're relying on secondary organisms. All right, and that brings us to our last component of the carbohydrates, and that would be something known as chitin. And this one is actually going to give us a pretty significant rigid structure for support when we start to take a look at some of the exoskeletons that might be found in. And we also find it in the cell walls of different forms of life. And one of the things that they've also gone and been able to establish is when you start to think about some of the sutures, the surgical sutures, in many cases it is composed of chitin-like material. So it gives you that flexibility and it allows for structure and rigidity. Okay, so that kind of wraps up our carbohydrates. So I'm going to stop here for a second on this.